Today we'll talk about the complementary genes. What are complementary genes? Complementary genes are two or more genes which interact to produce a particular phenotype. That means two or more than two genes, they are essential. They are necessary for the production of a particular phenotype. Those are called as complementary genes. So what are complementary genes? There are two or more genes which interact to produce a particular phenotype, to produce a particular phenotypic trait. They interact, the genes, two or more genes, they interact to produce a particular phenotypic trait. That means we can say that complementation between two genes means that both genes are necessary or essential for the production of a particular phenotype. That means if we'll say what are the important characteristics of the complementary genes, these are complementary genes are non-allelic genes. These are non-allelic genes. That means there are two or more than two different genes, and these complementary genes, as I told you that complementary genes are necessary or essential for the production of a particular phenotype, for the production of particular phenotypic trait. So these complementary genes, they occupy different gene loci. And when these are together in a dominant form, these complementary genes, they produce a particular phenotype. And if either of these genes is absent, the phenotype is not produced. So together in a dominant form, they produce the particular phenotype. Together in dominant form, they produce particular phenotype. Together in dominant form, they produce particular phenotype. That means these complementary genes, when they are present in a dominant form, they produce a particular phenotype. If there is absence of any of the complementary gene, then the particular phenotypic trait will not be produced. It means that the two complementary genes, they coordinate, they collaborate for the production of a particular phenotype trait. And when either of the complementary gene is absent, the particular phenotype trait will not get produced. These complementary genes were first reported by Batson and Punnett. First reported by Batson and Punnett in sweet pea Lathyrus odoratus. Lathyrus odoratus. These complementary genes are first reported by Batson and Punnett in sweet pea Lathyrus odoratus. This, in case of sweet pea, we have flower color. It is controlled by complementary genes. The flower color is purple. If dominant alleles of both the genes C and P are present, when the dominant alleles of gene C and gene C are present, then the flower color in case of sweet pea is purple. If either of these is absent, then the flower color is white. That means in case of a flower color in case of sweet pea, the genotype would be purple flower color, That means the genotype is capital C, small c, capital P, small p. Or genotype can be capital C, capital P, capital P. Or genotype can be capital C, small c, capital P, capital P. Or genotype can be capital C, capital P, and small p. That means when both the dominant alleles capital C and small c are present, then the flower color is purple. Same is the case, capital C dominant allele, capital C and capital P present, flower color purple. Same is the case, capital C, capital P present, dominant color, flower color purple. The flower color is white in case of white flower color. White flower color in case of this is white flower color, capital C, small p, flower color is white. Or flower color can be white, small c, capital P, that is flower color is white. That means in case of sweet pea, the flower color is purple when both the dominant alleles of a gene are present. It means that it has been found that this gene C, it synthesizes an enzyme C, and this enzyme it converts the raw material, precursor A, raw material, it's called as precursor A into chromogen. Then this chromogen is converted, it is changed into purple pigment, anthocyanin. It is converted into purple pigment, anthocyanin, by the product of gene P. We have got a gene P, and this dominant form of gene, and this will synthesize enzyme P, and this enzyme P will convert the chromogen into the purple pigment. That means how this purple color appears in the sweet pea, Lathyrus odoratus. When both the dominant alleles of a gene are present, gene C, it will synthesize enzyme C that will convert the raw material into chromogen, and chromogen is converted into the anthocyanin purple pigment in flower by the product of the gene P. This is gene P, gene C. So we can say that these two genes dominate together in a dominant form. That means gene C and gene P, they interact to produce the particular phenotype. When they are present together, they interact to produce a particular phenotype. If this gene, dominant form of this gene is absent, or if the dominant form of this gene P is absent, then the particular phenotype does not appear. That means in case of sweet pea, if dominant form of gene C is absent, then flower color is white. If the dominant form of gene P is absent, then the flower color is also white. But if both these genes, if the dominant form of both the genes, gene C and gene P, are present, then the purple color of the flower would be in the sweet pea. So it means that these two dominant complementary genes, they have coordinated, they have collaborated with each other to produce a particular phenotype. Now this, how we can explain it? We'll explain it by means of a cross. Batson and Punnett crossed. How we can explain it? Explanation. This Batson and Punnett, they crossed white flower sweet pea plants with the another white variety, white flower varieties pea plants. That means parents were parents. These are white flower sweet pea plant, and it was crossed with another white flower sweet pea plant, white flower sweet pea plant, Lathyrus odoratus. Batson and Punnett crossed two white flower varieties of sweet pea plant. This is their phenotype. Obviously genotype is, this is capital C, obviously small p. One dominant allele is absent. It means that the flower color is white. Same is the case. We have small c. Same is the case here. Only one dominant allele of a gene is present. Therefore, the flower color is white. If both the dominant alleles of a gene is present, then the flower color would be purple. This was genotype. Then we'll take gametes. Gametes will be capital C and small p, capital C and small p. Here also gametes would be small c and capital P and small c capital P. Two types of gametes. Then F1 generation is, we have F1 generation. We have capital C and small c, capital P and small p. So here dominant alleles of both the genes are present. That means the flower color is purple. So purple flower sweet pea plant. So this is purple flower sweet pea plant because both the dominant alleles are present, C as well as P. Both the complementary genes are present for the production of particular phenotype trait. This was F1 generation. Then we'll go for the selfing of F1 generation. So it is like this: capital C, capital P, small p. It will be crossed with capital C, capital P, small p. Obviously, what are the gametes? Gametes, or here gametes would be capital C and capital P, capital C and small p. Same is the case, small c and capital P, and small c and small p. 1 gamete, 2, 3 and 4. Same as the case, we have gametes capital C and capital P, capital C and small p. Then we have small c, then we have small c and capital P, and small c and small p. These are 4 different types of gametes. Then we'll explain it with the help of a checkerboard, a Punnett square. We have, these are gametes, male gamete, female gamete. These are gametes. This would be our F2 generation. So we have capital C, this gamete, capital P, capital C, small p, and small c. Then we have small c and capital P, and small c and small p. We have one, same as the case here, gametes capital C, capital P, capital C, small. Then we have small c and capital P, small c and small p. Then we can make the fusion of the gametes will take place. These are male gametes, these are female gametes, and fertilization, random fusion of the gametes to get the F2 generation. So we have capital C, capital C, capital P and capital P. So this would be purple because this is purple flower sweet pea plant. Then we have capital C, capital C, capital P, small p. This would be also purple flower sweet pea plant. Then we have capital C, small c, capital P, capital P. That means both dominant alleles of a gene are present, capital C and capital P. That means it is a sweet pea with purple flowers. Then we have capital C, small c, capital P, small p. This is also a purple flower sweet pea plant. Then we have capital C and capital P, small p. This is also purple flower sweet pea plant. This is capital C, small p. As one dominant allele of a gene is absent, that means we have small p, not capital P. Therefore, the sweet pea plant would be having white flower sweet pea plant. Then we have capital C, small c, capital P and small p. This is also purple flower sweet pea plant because dominant alleles of both the genes are present. Then we have capital C, and small c, small p and small p. This is white flower sweet pea plant. And then we'll take here, this is capital C, small c, this is capital P. This is purple flower sweet pea plant. Then we have here, this is capital C, small c, and capital P, small p. This is purple flower sweet pea plant. Then we have small c, and capital P. This is white flower sweet pea plant, because only one dominant allele is absent, one dominant allele is present, another dominant allele is absent. So white flower sweet pea plant. We have small c, then capital P, small p. This is also white flower sweet pea plant. Then we have here capital C, small c, and we have capital P, small p. This is purple flower sweet pea plant. This will take capital C, small c, and small p. This is white flower sweet pea plant, white flower, because only one dominant allele is present, other dominant allele absent. Then we have, this is small c, and capital P, small p. That means it will be white, because one dominant allele is absent. Then we have small c, and small p, it would be also white. I will explain. Ratio. This is the complementary gene action in case of sweet pea Lathyrus odoratus. So we'll have phenotype purple flower sweet pea plant is to white flower sweet pea plant. So we have purple flower sweet pea plant and white flower sweet pea plant. How many purple flower sweet pea plants we have? This is 1, 2, 3, 4, 5, 6, 7, 8, and 9. We have 9 purple flower sweet pea plant and 7 white flower pea plants. We have 1, 2, 3, 4, 5, 6, 7. We have 7 white flower sweet pea plant. So the ratio is 9:7. 9:7. The presence of purple flowers. The presence of purple flowers shows that the two genes have interacted to produce a particular phenotype out of 16 in case of F2 generation. In case of F2 generation, out of 16 we have 9 purple flower plants, and this indicates complementary gene action. This indicates complementary gene action. So in case of complementary genes, the normal dihybrid ratio 9:3:3:1. This normal dihybrid ratio gets modified to 9:7 in case of complementary genes, in case of complementary gene action. So we can say that the dihybrid ratio, normal dihybrid ratio, that is 9:3:3:1, that gets modified to 9:7 in case of complementary gene action in case of sweet pea Lathyrus odoratus. Thanks.